how are we going to dim a little bit the lights? No. This should be okay. Okay, so good morning again. My name is Chaya Kalheim. I'm from ELSEC, and I'm coming from the Faculty of Medicine at Ein Kerem, where I work and where I have my lab. And my lab is specialized in developmental neurobiology. So in the frame of your very, very big course in physiology, I think this is the name, right? We are devoting 4 hours to developmental neurobiology. That is, in different words, the formation or the generation of complexity. Because we all argue, and this is, I guess, why you are here, the idea is that we are all working with the most complex system in our body, right? Maybe one of the most amazing ones. But you will be surprised how complicated. Okay, let us wait for everybody. Many people, more than last year. So the idea is trying to understand how this complexity is generated. So undoubtedly, you will bear with me. In 4 hours, I will not be able to do this. I mean, I will not be able to convey you all the details of how the nervous system develops cellularly at the level of the tissue, and certainly not at the molecular level, which is the one we study in combination in the laboratory. But I'll try to give you a taste of some key processes. And what I'm going to do is the first hour is going to be basically an introduction, a general introduction of all those. Why don't I? Yeah, it should work. So I gave a few lectures yesterday, and I am out of voice. So what I would like to do is to give you, during the first hour, a very, very short introduction of the main processes that take place during development of the nervous system. How do we integrate the nervous system within the context of the development of the whole embryo? Because it's an integral part of the embryo. When you study nervous system here, as far as I am concerned, from many of my colleagues at the neurobiology department in Hadassah, at ELSEC here, the brain is the brain and nothing else exists. But this is not true. The brain is an integral part of our body. It is influenced by hormonal inputs. It influences behavior. It is influenced by your gut. Amazingly, this is one of the most popular topics now in biology in general. Have you heard of the microbiome? Okay, it is these millions of milliards of gut bacteria that are associated more and more, or are being associated very convincingly with behavior, with depression, with anxiety. And it appears that the gut, which we call normally the second brain, okay, and rightfully for many of us, influences all our higher mind functions. So I want you to look at the brain in terms of development as something which is completely integrative. The brain is part of your body, and the body is part of your brain. So with this, along this vein, we are going to begin our journey, little 4-hour journey. What do you see here? Let me translate for those of you who are not Hebrew speakers. Okay, so this is a fish, this is a salamander, this is a turtle, this is a chick, this is a rabbit, and finally, not surprisingly, also very ugly, this is a human being. Okay, so this is what we see here. This is called the Haeckel's principle, and I would like your input, your absolutely unbiased and naive input, to tell me, what do you see? Let me just point out that what we are looking at here is different stages of development of different species. Up in the upper part of the slide, what we see is early development, later development, and let's say a very late developmental period. Okay, anybody, please. right? Okay, so the first thing that we can see is that along evolution, the earlier stages or the earliest stages of development look very similar in a fish and in a human being. Okay, let us develop a little bit further this. Okay, and this idea means that there is evolutionary conservation, and we know now from many, many studies. Can I turn on this volume a little bit higher? How do you do this? Microphone volume, but it is not this microphone, it's this microphone. Okay, never mind. So we know now that basically there are many, many conserved genes during development of all the species. And I'm going to give you one example. There is a family of genes that is called Hox genes, H-O-X. These genes are composed of a sequence of DNA that is called the homeobox. This sequence of DNA appears to be conserved from Drosophila, you know, the fruit fly, till the human being. Now, these genes are localized along the chromosome in a certain sequence. I'm not going to detail because I can give you an entire course about this particular family of genes. There are many, many genes, like 30 Hox genes, and they are localized along the body. Now, what do they do in Drosophila? In Drosophila, there is a Hox code, a combination of different Hox genes along the neural axis, along the axis, in all the tissues of the body. And it appears that they define in the Drosophila the identity of different segments to become the head, to become the thorax, namely the trunk, the abdomen, and the tail of the fruit fly. Now, Nobel Prize many, many years ago was the finding of the conservation of the Hox genes along evolution. And it appears that we have also Hox genes. And let's go to the mouse, our next neighbor, for laboratory purposes. If in the mouse we misexpress a Hox gene that characterizes the area of the vertebra that gives rise to ribs, and we misexpress it in the neck, which doesn't have any ribs, we will obtain a neck containing ribs. So this means that also in higher mammals, in higher vertebrates, the function of the Hox genes has been conserved to determine the segmental identity of a certain body part. What are we? We are segmental animals, right? We have a head. Have you thought about it? We have a head. We have a neck, which is very different from your head and also different from your trunk. We have a tail, okay, which is very different in terms of the nervous system and its behavior and its nerves, etc. And we are segmentally organized. We have 2 forelimbs coming out from a certain part of the body and 2 hind limbs coming out from another part of the body. And this is determined by the Hox code. Okay, so the segmental identity of our bodies is determined, and what this tells us, this principle has been stipulated like 150 years ago. And now we know not only that it is true, but we know also the molecular underpinning of this principle. So this is something to take home about. You can read about it books and books and books, and I just chose to bring you a glimpse of it so that you can get an idea and think of this issue. Now, concerning the nervous system forms in these embryos. This is the spinal cord, this is the brain. So eventually in a fish, the brain is going to be very small, and in the human being it is going to be extremely well developed. This is called the process of cephalization. It is a novel process that took place during vertebrate development, during the transition from aquatic behavior to terrestrial behavior, and I will deal with it, I hope, next hour. Okay, when we'll talk about the development of the brain, peripheral nervous system, and the skeleton that covers the brain, which is something extremely important and relevant to the development of the nervous system. What else can we see here? What characterizes fish? Do you know what this is? I can spend 2 hours on this slide, but this is going to be the last point. What is this? Gills. Perfect. So what is the function of gills in fish? Breathing. Okay, so the entire transport of oxygen from the water to the body takes place through these structures, which are quite complicated. They have all the 3 germ layers: ectoderm, mesoderm, endoderm. Okay, if you have heard of it. I don't know what is, I forgot to ask about your background, but anyway. So, have you noticed that we also have gills when we are embryos? So it appears that there is an evolutionary conservation during early development which goes beyond any imaginable thing. So human embryos have gills. It is not called gills, it is called branchial arches or pharyngeal pouches. It depends if you look at it from the mesoderm point of view, from the ectodermal point of view, and they are not going to persist. I don't have gills any longer. But I have 3 different ossicles, 3 different bones in my ear, the stapes, the, how are they called? Forgot. All the 3 ossicles in your inner ear, they come from the gills. Part of your thyroid comes from the gills, of your parathyroid, and other different endocrine organs, they develop in our body from the gills. So in this sense, the gills evolutionarily exist in the very early embryo, but have been transformed to generate different derivatives in the vertebrates. Gills in the embryo are never functional, in none of the embryos. Of course, in the fish, when these fish are out in the water, they will need immediately to use the gills, pretty much as larvae of a frog embryo. Okay? The moment they are released into a freely moving system, they will use the gills. In our case, none of us has to perform any respiration functions until we don't see the outer world, because the placenta with the umbilical veins and arteries does the job. Okay, we are placental animals. So the functions have been changed enormously throughout evolution. So as I said, Ken, Vakasha. Sorry, please. No, just ask. Ask everything, okay? I don't mind if we cover half of the topics that I intend to cover, right? So I prefer it to become a dialogue rather than to become, I mean, in any event, my expectations from a public like you, it's not like in medical school, that in embryology, all these future doctors have to cover a very tight curriculum. Many of you are physicists, computer people, few of you, I guess, are biologists. So it's a kind of treat, the cherry on the cake, these 2 courses. So you're going to keep it in mind, use it, forget it. So I prefer you to, okay, to enjoy it rather than to take it as a heavy stuff. Okay. Human embryos have to grow all the tissues, okay? Every tissue has to develop, and in order to develop, it needs oxygen and it needs nutrients. Now, nutrients and oxygen come from the placenta. Okay, in the embryo, you have an umbilical vein, 2 umbilical arteries. I mean, you have 2 and 2, but then 1 degenerates. And the placenta, which is the interface between the mother and the fetus, performs all the necessary tasks to oxygenate the embryonic tissues. So the embryo doesn't do anything. The embryo, what it does is it basically, from a certain point in development, when the mouth develops, it takes into the digestive system amniotic fluid, okay, water that we have in the belly. It takes out amniotic fluid. It releases amniotic fluid from your rectum and anus that develops in the embryo, and everything goes back and forth. And the same for the CSF of the central nervous system. It's a closed system, and then everything gets read out from the placenta. Okay, so the placenta is a kind of waste trash, but it also, without it, you cannot live. Okay, so women with placental problems have difficulties and many malformations of the embryos and so on and so forth. Okay. So this is just to put in context the idea that the nervous system is part of the entire development of the embryo. We will continue to develop this idea. And I would like now very, very briefly to go with you through very basic events or processes that affect embryonic development. So what we can see here, and these are also correct for the nervous system. So again, the point I want to make is that the nervous system is part of the organic development of the embryo and not something which is an outstanding process, certainly not at the beginning. So first of all, cell specification and phenotypic differentiation. A neuron or a progenitor, which comes from the ectodermal layer, we will see that in a minute, has to get an identity. This is the first thing it has to do. I have to be a neuron, and then I have to decide in a hierarchical manner which type of neuron am I going to become. A glutamatergic neuron, a GABAergic neuron, a cholinergic neuron. And this depends on my position throughout the body. Am I going to be in the cortex? Am I going to be, I don't know, in the amygdala? Am I going to be in the spinal cord? Am I going to be in the peripheral nervous system? And depending on my final position, I am going to take this decision. Then I have to decide, am I going to be a neuron or a glial cell? This is a second decision in the row. But this is called fate specification. Namely, how do I adopt a certain identity during development? This is true for a muscle cell too, and for a cardiac muscle cell, and for a gut cell, and for a pancreatic cell. Okay, so again, the fact of deciding whom I am going to be when I'm a grown-up cell is something which is a universal value. Okay? Next, cell proliferation and differential growth. We are born from one single cell, which is called the zygote, the fertilized egg, namely a female egg with a sperm that creates a molecularly, genetically distinct organism. Each one of us is different from your neighbor. Okay? So this cell, the first thing it has to do is to proliferate. It undergoes many, 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 many cycles of cell division. But for example, if you look at your gut, okay? Your gut has an esophagus, which is a tube. But then comes the stomach, which is not a tube. It's a ball like that. It's like 5-6 times bigger than your esophagus. And then comes your small intestine, okay, which is like 1.5 meters long. So this is differential growth. Everything has begun in the gut, pretty much like in the nervous system, as a single small tube. The nervous system is a little tube. And then you get a brain, and then you get a spinal cord. Very, very different one from each other. Okay? Many more cells in the brain than in the spinal cord. However, neurons you have in your spinal cord pretty much as you have in your gut. Think about it. Okay? So cell proliferation is a key event in development, and also differential growth. Namely, certain areas in your system, call it nervous system, call it as you wish, will proliferate more than other areas. And this is called differential proliferation or differential growth. Then epithelial to mesenchymal transition or mesenchymoepithelial transitions. I will talk about it when I talk about the peripheral nervous system. We will begin with it a little bit in the next hour and then the next week. Most of the cells in your body are created as an epithelium. Anybody knows what an epithelium is? Speak up. It's a layer of cells. So an epithelium is basically defined as a layer of cells. These are the nuclei. A layer of cells that are a layer of cells that are bound by what we call adherence junctions, tight junctions, a basement membrane, apical domains which are tight, namely like soldiers that are, arranged one next to the other and bound to each other very tightly. They cannot move, okay? And they are usually barriers for transport of molecules and stuff like that. And this is called an epithelium. But during development, you know that cells have to migrate. During development, many times, and this is the case of the neural crest that I'm going to talk about it next week, these cells arise in the spinal cord, in what we call the neural tube, and then have to migrate throughout your entire body to innervate the gut, to give rise to dorsal root ganglia, sympathetic ganglia, chromaffin cells, and so on and so forth, all of them outside the CNS. And to do so, they have to become motile. And to become motile, they undergo a process that is called epithelial to mesenchymal transition, okay, by which these cells lose apical junctions, tight junctions, basement membrane, and so on and so forth, and they become motile. They become stellate. And then they can move through actomyosin, through Rho GTPases, and all kinds of molecules that are devoted to the ability of enabling this cell to move throughout the different tissues of the embryo. And then they migrate by different cues, which are stereotypic, of which we know many, many of them. So a lot of cellular and molecular biology all around. Cell migration, of course, can...
I don't understand, they migrate and they become other cells, or they just let other cells migrate?
No, they migrate as progenitors, namely as precursors of what they are going to become, and then when they stop migrating, they organize into tissues. One example I told you, the neural crest, we will speak about it, you don't have to know this name, is a transient progenitor population. These cells, the hallmark of these cells is to migrate, to invade adjacent tissues, and then to stop migrating, and then organize into ganglia, into aggregates of neurons and glia. Okay, so migration is the means by which an embryonic cell can disperse throughout the embryo to finally differentiate into a certain fate, to give rise to a certain phenotype, which is different from its origin. Okay, this is something you have to understand. The timeline of development means that at every day you are a different thing. You begin as a progenitor, but then when you migrate, you have different molecular characteristics than those that you had when you were an epithelium. And then when you stop migrating and begin differentiating, you are a third thing, which is different from what you were when you were migrating and different from the original source. So in development, everything changes every minute.
Is there already epithelium cells that they go back to the progenitor?
No. The epithelial to mesenchymal conversion is associated with the change in fate of the cells. Okay? And when they aggregate into a ganglion, for example, outside the CNS, they don't become epithelial. Some of them can become epithelial in other cases, but then they are different in terms of their identity. So you have to understand that embryonic development is a permanent change in the cellular and molecular composition of your cell and also in your identity. Okay, you begin as a progenitor, you end up as a neuron, as a glial cell, as a liver cell, and so on. Okay? Every step means a change. So, tissular fusions, we are not going to talk about it. This happens a lot in the development of, for example, the intestine, budding as well, organ rotations, and something which is called apoptosis is extremely important in the nervous system. It has been basically discovered. Anybody knows what apoptosis is? Cell death. It's one form of cell death. Programmed cell death. Okay, even more. Okay, even fancier. Programmed cell death or apoptosis means that during development, our system creates many, many more cells than those that will persist during adulthood. And there is then a mechanism that begins to be applied on the system, normally after innervation of the target organ, that says, okay, 100 cells underwent synaptogenesis, but the size of the target does not maintain 100 synapses. This is too much. Why? Because we don't have enough nutrients to maintain 100 cells alive. We have 450 cells. So then there is a process of retrograde cellular death which will generate the death of those neurons that innervate the target that are superfluous. And we'll see it in a few minutes. So apoptosis, or programmed cell death, is a very, very important process without which we wouldn't be normal. Too many synapses, too many cells innervating something means abnormal. So it turned out that apoptosis takes place as a normal part of development. So cell death is part of our normal development. There is also, of course, cell death taking place every minute in our brain as we age. This is not necessarily apoptosis. Okay. You had a question first, right? Of course not. There are like 10 different methods of cell death. There is necrosis and necroptosis, and this has been recognized initially, and it is still the major type of cell death in the nervous system during development. It is target dependent, okay, and it has been recognized initially in the nervous system, okay, basically by the group of Victor Hamburger and also by Rita Levi-Montalcini. Maybe we'll talk about it a little bit later. But then it appears that most of the cells in our body undergo apoptosis to match somehow the target organs. However, apoptosis, for example, has not been detected in cartilage. This is one of the only systems, cartilage, one of the only systems in which we do not detect really an apoptotic type of death. But most of the systems in our body undergo apoptosis. Okay. Before birth and after too. It depends on where we are talking. There are systems in our body which developed prenatally, most of your spinal cord. So apoptosis occurs quite early, mid-gestation, even earlier. Very close to the time of target innervation, synaptogenesis. But there are tissues, for example, your cerebellum, which greatly develops postnatally. So their apoptosis is going to go beyond birth. So it depends on the system we are talking about.
So we know what is the advantage in having two more cells and kill them instead of just enriching the spinal cord?
Yeah, there is. It's not a okay, it's a very tricky question, because in biology you basically don't ask, you can speculate about advantages and disadvantages, okay? In biology you just figure out what is the mechanism for something, but not what is the reason for something. The reason is something philosophical, and I see you have a kippah, so you can explain that. So the idea basically is that at the beginning there is no connection with the target organ, because, for example, your limb bud, which is going to need a lot of neurons to innervate it, okay, has not appeared yet. And the flank, which will need less neurons to innervate it because the size of the target organ is much smaller, it doesn't exist at the beginning in any connection with the spinal cord where the neurons are generated. So at the beginning the system generates neurons in a completely autonomic way. But then when the neurons grow out axons and there is synaptogenesis, then there is a feeling that here I will need many neurons. So there is almost no cell death in the brachial area of the spinal cord because we need all of them. But there is a lot of cell death in this area of the axis, because then the neurons realize that there is not sufficient target. And sufficient target means sufficient trophic support. Molecules that are produced by the target, like nerve growth factor, brain-derived neurotrophic factor, etc., which have to go retrogradely towards the cell soma, towards the neuronal body, and say, you will be staying alive. Not enough trophic factors, not enough survival, so you die. This is what we call the neurotrophic hypothesis. I'll show you just a brief thing about that. Okay, so now the nervous system. What do we see here? Anybody knows what endoderm, mesoderm, and ectoderm are? The three germ layers of the embryo? Okay, so the embryo at the beginning grows and proliferates and is like a ball full of cells, okay, that we call morula. But then it flattens and it creates layers, and it basically creates 3 layers. This is like 5 hours of teaching, what I have just said now in 2 words. Okay, so it creates 3 layers, And these three layers can be seen, for example, here. This is the ectoderm, this is the mesoderm, and this is the endoderm. Now the ectoderm will give rise to your skin and to the entire nervous system. The mesoderm will generate muscle, vertebra, blood cells, blood vessels, dermis, namely the subectodermal portion, fat, adipose tissue, and so on and so forth. And the endoderm, this, will generate the gut. And everything will fold on itself to give rise to a cylindrical structure. At the beginning, we are all flat animals, but then we fold. You see that we are like a baguette? Okay, have you noticed that we are closed animals? Okay, this happens if anybody has a sheet of paper. You don't use them? So we are all developing like that, 3 layers. Imagine you have 3 sheets of paper, but then we do this. Okay? So the endoderm, which is here, will close a tube, and this tube will become your gut. And then it will develop into a mouth, an esophagus, the intestine, stomach, etc. Okay, so this is called the process of embryonic folding. So what you can see here is an embryo which hasn't folded yet. It has nothing to do with the nervous system, by the way. But the nervous system is here. So what we have here is the ectoderm, which is this layer here, and the endoderm. The mesoderm is not there yet. Now let us see a little bit a later stage. Now what you see is the following. The nervous system develops from ectoderm, from the upper layer. And it undergoes a process of induction, of initial identity formation, which is called neural induction, by which the ectoderm, which is normally going to give rise to epidermis, to the skin, is going to thicken. Can you see this? Can you see this? This is contiguous with the ectoderm. This is one layer. But in the center, in the middle, imagine the area of your spinal cord, okay? We are looking at this area. In the middle, these cells are going to become thicker. So this is one single cell, and this is one single cell. But this cell is thicker, and it is called pseudostratified epithelium. If we have time, I'll tell you about that. Now, once this happens, this is called the neuroectoderm. Namely, once the neural induction took place, these cells are destined to become nervous tissue, and nothing else. They cannot become epidermis any longer. So this is the dynamics of development. Once you became neural ectoderm, you don't follow an epidermal fate any longer. So once this has become a neural ectoderm, it will begin folding. Can you see that? And it will create a groove in the middle, and finally it will close and will generate a tube. And this is called the neural tube. The neural tube will generate 3 things: the spinal cord, the entire brain, and also the entire peripheral nervous system via a group of transient cells called the neural crest cells. So the entire nervous system is going to derive from this simple tube that runs from the rostral part of the embryo till the tail. Okay? I'm not going to tell you how it develops along this axis because, again, this is a matter of an entire course. So this is the idea. Another thing to remember here is that the neural tube doesn't develop on its own. In terms of what it's going to generate, it has intimate connections with the ectoderm, which gives him a lot of factors for development, with the mesoderm. These are going to be called the somites. This will generate vertebra and muscle. With this structure here, very, very important structure, that is called the notochord. It's like a road, makel, okay, going through the entire embryo up to a certain area. We'll deal with it in the next hour. Okay? And the notochord will provide a very important factor that will generate the diversity of cells in the neural tube. So this is the frame on which we are going to work next. You can see that also in a kind of schematic drawing. This is the neural ectoderm. This will become epidermis. This is already neural. It expresses encadherin and not epithelial cadherin. It will fold, and then it will form a tube, and the ectoderm, which will become epidermis, will regenerate above it. So this is the general idea. Now, this is not the whole story. I'm not going to tell it to you, because this happens up to the umbilical area. And then the rest of our spinal cord is generated a little bit differently. It's called secondary neurulation. And it is very similar to what happens in fish throughout the entire axis. Secondary neurulation, just keep it in mind. I'm not going to tell you how it happens. It's a little bit more complicated. Many people, most of the people, talk about this mechanism of invagination and closing of the tube. So let's stop it there. But you should know that this is not the only story. Okay, now, what happens? The neural tube, when it forms, is homogeneous. The dorsal part and the ventral part, everything is the same. It is naive. Okay, all the cells are the same. Now we have to begin creating motoneurons, interneurons, and sensory cells of different types, namely cells which will sense the periphery, integrate it, and form synapses with the muscle so that I can move in a reflex manner, even without my brain, move my foot or whatever. And this is what we call pattern formation. Do you see these gradients of green and shades of violet? This is called pattern formation. How does this happen? How do we turn a homogeneous epithelium into a segregated population of different subsets of cells? Any idea? Yeah, I already gave you a hint to that, and this is obviously the main idea. So we know that, and this is the general rule, that to pattern the dorsal portion, this is dorsal, this is ventral, okay? To pattern the dorsal portion of the spinal cord, let's stick to the spinal cord, it's the same in the brain, because everything is a tube at this point. We need factors coming from the epidermis as well as from the dorsalmost portion of the tube. And to pattern the ventral portion of the tube, we need factors that come from the notochord, this road whose name I just mentioned, and from the ventralmost part of the tube. So this is called the morphogen hypothesis, namely, the more concentrated or the higher concentration of the morphogen will come closest to the origin, to the source of the factor. And the cells which are closer will get a higher concentration. And the cells which are farther apart will get a lower concentration because these factors diffuse. So by law of diffusion, you will get exposed to different concentrations. And the difference in concentration of the factor will determine who you will be. Okay? And the same for the dorsal factors. The more concentrated the factor is here, this cell will be exposed to a higher concentration than this cell. And based on the different concentration, you will become a different type of neuron. Now, this theory underwent a lot of modifications, namely, it is not only, and a lot of mathematical modeling for those of you who are aficionados of that. So it appears that it's not only the absolute concentration of ligand to which you are exposed, but also another value, give me? What?
How much you're exposed to the alveolar.
Okay, how much you are exposed to the antagonistic. We'll talk about it in a second. And change, internal. Give me the key point of development. Time. So time is extremely crucial here. So it appears that if I give a certain naive cell a very low dose of ligand, but I expose the cell for a very long time to this dose, the cell will know how to integrate this low concentration over time to become equivalent to a higher concentration over a shorter time period. So it appears that the same, and I'll show you a little bit of data from our own studies next week. So basically, time and concentration are two equivalent values in the determination of the fate of a certain cell. Okay, so now let us talk about the inducer, the morphogen, the factor. Okay, where are we? Okay, still like 5 minutes to go. So let us look, let me do it from here because I have to change now frequently. Let us look at the neural tube. This is the neural tube. Okay, and this is a marker that is called, this is an area that is called the floor plate. And now we are marking, we have stained with a marker that is a floor plate specific marker. Never mind the name, it's not important. And here you have the notochord, right? This is a cross section. We took the baguette and we sliced it. So this is what you can see. Now, what do we see here? We have added an additional notochord. This is microsurgery. In the chick embryo, you can do this very easily. Because the embryo sits in the egg, you can open the egg, you can do microsurgery, you close the egg with sellotape, you put it back in the incubator, and you can hatch it. It's not easy, but it's doable. So many experiments in embryology have been done using chick embryos, which are also the model system we use in our lab. So look at this. Here, people have grafted ectopically, it's not normal, another notochord. There is never a notochord in this area. But in addition to the normal one, they have added another notochord. What happened here? What is the outcome? Two floor plates, correct. So what we see here is that adjacent to the notochord, in an ectopic way, due to the high concentration of a substance that the notochord produces, we have induced within a naive tissue another notochord. In other words, we have ventralized the neural tube. Because this is ventral, this is dorsal, we have taken the dorsal part and we have ventralized it. We have changed its fate. Okay? This is doable in embryogenesis. Now what have we done here? What we have done is we have added two notochords in lateral position vis-a-vis the midline, and you have developed two floor plates, one adjacent to the other. This means, first, that the system is plastic, that at this point in development you can change the fate. Second, it means that the notochord provides a factor able to change the fate of cells. Namely that the change in fate is inducible via extrinsic factors. Right? Are we okay with these interpretations? Now let us look with a motoneuron marker. This is a molecule, never mind the name. These are motoneurons. And these are the fibers of the motoneurons which go to the periphery and will innervate your muscles. Okay? This is the floor plate. This is the notochord. These are the dorsal root ganglia. They also stain with the same marker, but they are not relevant right now. So using this motoneuron marker, and this is very important too, look at this intermediate zone. These here are ventral interneurons. These are interneurons which communicate between different motoneurons and interneurons that are localized in the dorsal portion. They are called ventral interneurons of type 3, never mind. So we are not staining for them, so you see them as a negative. Okay? So what are we doing here? We look at the distance from the inducer, okay? And you see that to become a floor plate, you need the highest concentration of the inducer. To become V3 interneurons, and we could have stained for that, but it is not done, you need an intermediate dosage of the ligand, and to become a motoneuron, you need a lower concentration, a relatively lower concentration, which is absolutely a function of the distance from the inducer, which is the notochord. Now again, if we graft the notochord in this area, look, the motoneurons, these are ectopic, abnormal motoneurons, will develop there. They will not develop close to the notochord as the floor plate did, as we saw with the floor plate marker. They will be always a distance between the grafted notochord and the motoneurons, pretty much like under normal development. And this means that to become a motoneuron, you will have to be exposed to a lower concentration of ligand. Ken? There is. Yeah, it's not obvious, but there is. Because we are not here staining for motoneurons. This is like junk, what you see here. Okay, it's not, there is a floor plate and there are V3 interneurons as well. Okay, this is a very old slide by other people. Okay, never mind, let's continue. And the same, of course, is correct with floor plate markers. Let's continue. Which is the factor? The factor is called sonic hedgehog. It's a factor which has been discovered in Drosophila. It's a very, very important factor. It does everything. Something you have to remember. In development, there are like 7 to 10 factors which create the entire embryo. So how can we become what we are with only like 8 to 10 factors? It's because in a contextual manner, each one of the factors is able to elicit a network, a cascade of transcriptional factors downstream of it, which creates a geometrical tour, something which is amazingly complicated. And that in each different tissue and at each different time, each of these factors will act differently. At a certain time it will promote proliferation, later on it will promote differentiation. So they are reiterated in activities. They will do many things and different from each other at different times. So with very few factors you can create an embryo. And sonic is one of them. So sonic hedgehog is a factor, this is the tube, which is expressed at the beginning very, very early only in the notochord. And then it becomes expressed in the floor plate once the floor plate is created. So when you stain for sonic hedgehog, this is what you get. Floor plate produces sonic, notochord produces sonic. This is the origin of this molecule. Now, this is one of the most complicated molecules, and if you see it, of course, throughout the embryo, you see it in the notochord from tail to, not head, but up to here. So this is sonic. How does sonic act? I'm not aiming at giving you, I'll finish this one and we'll take a break, okay? I'm not aiming at you understanding everything here, not even far from it, but just give you a glimpse of the complexity. So sonic basically acts within an organelle of the cell that is called the cilium. Each one of our cells, including nerve cells, contains cilia. Cilia is, if you look at a cell, is like a little protuberance that comes out from the cell, like a tiny hair, very, very small. But it is an organelle in which many signal transduction processes take place. Wind signaling, BMP signaling, sonic signaling. Now what happens? It appears that sonic works with two receptors. One of them is called smoothened and the other one is called patched. So once sonic comes, okay, this is sonic, it will first contact patched, and then patched will leave the cilium and smoothen will enter the cilium. Sonic will then create a complex with both receptors, and then smoothen will transmit the activity of sonic into the cell. By transmitting the activity of sonic, it will act on three different factors which are called Gli factors. Gli1, Gli2, Gli3. In the absence of sonic, these Gli factors go to the proteasome, which is another organelle in the cell, for degradation. Bye-bye, ciao, to the trash. But in the presence of sonic hedgehog, these Gli factors are going to go to the nucleus. They will survive. and they enter the nucleus where they will elicit the transcription of target genes of the pathway. And these target genes are different in different contexts. In muscle they are A, in nervous system they are B, in the gut they are C, in the pancreas they are D. Okay, this is the complexity of the system. Now sonic has many friends and many enemies. It has 3 friends which are co-receptors that are called GAS1, KIDO, and BOC. These are transmembrane receptors. Don't forget completely about the names. It's not important. Everything comes from Drosophila. Okay, in Drosophila they have so many funny names, and we have adopted them for the vertebrate system. So GAS, KIDO, and BOC, they are friends of sonic. Wherever GAS, KIDO, and BOC exist, they create a complex with sonic and they stabilize sonic activity. So sonic activity is going to be prolonged and is going to be more efficient. But sonic has enemies. One of the enemies of sonic is called hedgehog interacting protein. And what hedgehog does is it binds to sonic, creates a complex, but has no ability to transduce the signal. So it acts like a buffer. It acts like a sink. I bind sonic and I do nothing with it. So basically the activity of hedgehog interacting protein is to remove sonic from the system. Okay, so everything is checks and balances. And every system, guys, has everything. In the neural tube you have hedgehog interacting protein, you have KIDO, GAS, and BOC. You have everything, and this is only a partial list. And the GLIs, GLI1 and GLI2 are activators of sonic. GLI3 is an inhibitor of sonic. So the end product of the activity of sonic is not only the concentration and not only the time, but also the mix of all the enemies and friends that sonic recruits in its journey from the notochord towards the target cells. Okay, and this is, I'll answer questions and we'll take a break. And this is basically the matter of like 30 years of research, and it is still far from being completely understood. Ken, bבקשה. Nobody knows. Most of the cells express them in a very dynamic fashion. Nobody knows. The context. If you are in the nervous system, you will have a certain set of transcription factors that will become activated. And if you are a gut cell, you will end up, because development, again, is not only about the expression de novo of certain transcription factors, it is mainly about the shutoff of the activity of irrelevant transcription factors. At this stage, no. This is a stage in which already the system is working, so pretty fast they become different from each other. Okay, we do not know whether at this very early stage everything is already the same. I mean, to say same and to say different means that you need markers. I am not naive. I don't think we have all the possible markers. We have markers. For example, when I did my postdoc, we had markers of differentiation, which is the last stage of the determination of a cell, when it becomes already active. Now we have markers of specification. Maybe, okay, in 10 years we are going to have, I don't know, epigenetic markers, which will mark even earlier stages of development that we are unable to detect now. So maybe heterogeneity can be detected earlier than now. Additional questions? Okay, so let's take a break and then we convene. We'll talk about it in the next hour.
Yeah, I mean the
other factor is called BMD.
Oh, okay.
Which, okay, we'll talk about it in a minute. שהעובר לא כזה אסטרונאוט, הוא פרזיט.
אוקיי, ואז בדיוק שפותחים את הגור,
נכון,
אז
אז מה
הם
קורה?
מתנוונים? זה העידן של האמא.
הם לא רק מתנוודים, את מעיפה אותם.
כן, אבל אני מניחה שיש חלקים שהם...
בוודאי מתנוודים מהר מאוד. סוגרת, את עושה קלאמר, סותמת. בלידה, היית בלידה? לא. זה מאוד נחמד. מה שקורה זה שהעובר יוצא, אוקיי, והעובר מחובר לחבל הסבור דרך כל הווסטה, זה בעיקר אומביליקלי, אוקיי? מה שאת עושה, קרוב לפה, מפה זה יוצא, אוקיי? בערך במרחק של סנטימטר, את שמה קלמרה, סותמת. הדבר הזה גם גורם לשינויים מיידיים בלחץ החמצן בתוך העובר, מוריד את לחץ החמצן, פתאום לא מקבל חמצן יותר. ואז זה מחייב את הריאות של העובר לעשות את זה.
לנשום
מיד, אחרי.
זה שפותחים משהו כזה?
בטח. העובר, את יודעת שהריאות של העובר הן מלאות במים, מלאות במי שפיר. העובר לא נושם.
אז כאילו,
העובר עושה תנועות של נשימה, אבל הריאות מלאות במים, הן לא עושות שום דבר
אז
פונקציונלי.
יש לי לידה, ואז יש לו מים מתחלות,
בשתי צורות, א' דרך הפה ואף, לפעמים המיילדת גם כן שואבת קצת, המיילדת שואבת קצת מעל וזה, ודרך דיפוזיה לרקמות.
אז
בעצם הלידה מבחינת הריאות של התינוק זה מעבר ודיספוזל של מצב שהריאות חצי מלאות בנוזל אמניוטי לריאות שמתמלאות באוויר. כי העובר, מה הוא עושה כשפותחים לו את זה, כשסותמים פה, הוא מתחיל לנשום. למה לא כשהוא מקבל את כל סטורציית החמצן שהוא צריך? אמרתי לך, הוא פרזיט. This is exactly what we are going to deal with at the beginning of the lecture. Okay, if anything is not clear, then I'll explain it to you. I went beyond the time and we'll look at it.
They said that time is an important factor, and that the shell is exposed to slow precipitation, but for a long time in the face the precipitation is it results in the same thing as
More or less.
More or less, okay.
Yeah. All this is mathematical modeling. It's very difficult in biological terms to do this. You can do it in culture, but you cannot do it in the end. You can do like mimicry, like putting a cell in culture and then giving refreshing concentration, for example, science modeling. So within the limits of the credibility of an in vitro experiment, this is what it is. It depends on the species. For example, in the chicken embryo, this is a matter of 1.5 days. 1.5, 2 days, more or less. In the mouse embryo, it can be a matter of 3 days. In the human embryo, the final point is 8.5 days, closest to 15, okay, after fertilization, and it's a matter of 10 days. By the become cells are, if you have a cell that is like that, and the cell suddenly becomes like that, okay, so then a structure which is flat will become, now you can ask what turns this cell to become cuboidal, from cuboidal to conical. It seems that what happens is that the embryo, you know, the embryo is not free. Okay, it is bound by extra-embryonic membranes within the placenta or even in the egg. So we believe that if there are longitudinal forces of growth, cell division, this will create a kind of groove. Like, for example, imagine that you have something like that, you play this, and you will have automatically a groove, okay? Okay, so this will be, so mechanical force will be a driving force for different motor molecules in the cell to change position.
Like a constriction?
Exactly.
Just one last
Exactement, monsieur.
I think that התיאור של אקל,
כן.
נראה לי that we learned with אוריגת, in the developmental biology, שהוא לא נכון.
In every model, there are things which are correct, and there are things which are not correct.
But that he didn't see it, that he just drew it. We don't know that?
This I don't know. But let me point out that those things that I pointed out are
yeah, for sure.
So is it clear? Is it too complicated, too simple? Well, I guess that everybody will get it differently.
I have like a developmental course.
Okay, so let's continue with this idea of the gradient and how basically, and I was asked the next, exactly what I'm going to present to you now, I was asked during the break, so I hope to meet these questions, and if not, just tell me. Okay, so the idea is how basically the final fates are established. Is everything a matter of morphogens coming from dorsal or ventral? Let me just say that the ventral morphogen is sonic hedgehog, and the dorsal morphogen, which comes from this area here called the roof plate, is BMP. It's called bone morphogenetic protein 4. This is a molecule that has been initially discovered to be a bone-forming protein, also a secreted molecule, but later on has been found to be widespread in your entire body and in the nervous system to play a very, very important role. So they are two antagonistic molecules playing a role in patterning the dorsoventral extent of the neural tube, sonic hedgehog from below and BMP from above. So how does this take place? So here we are talking again about a gradient, very concentrated in the ventral domain and more diluted as we go to the upper part. So if we look, for example, at the expression of different genes, for us, these colors now represent different genes. We see a gene, a blue one, whose extent of expression is ventral. This one is broader, and this one is even longer. So we have three different genes with three different extents of expression. None of them reaches really the dorsal area. But now, what we see is that there are other genes which are induced by the dorsal inducer here, and whose expression is extinguished as we go towards the ventral part. Why is this so? Because on the one hand, sonic will induce the expression of the blue, violet, and yellow, but will inhibit the red, violet, and green. Okay, so dorsal genes are inhibited by sonic, and ventral genes are induced by sonic. So how do we refine a territory out of this very coarse initial pattern of expression? So let us look at this area here, okay? And let us look at this area across the expression of all the genes in the row. So at the beginning, this area will look like that. If we combine all the colors, it will be very red here, very blue here, and have a very fluid type of really no boundary. Okay, but later on something happens, and this is something which is intrinsic to the system and independent of the morphogen. Namely, what sonic has done, it has done. Sonic has induced these ones and repressed the ventral expression of these ones. Pretty much like BMP does it reciprocally for the dorsal area. But now you can remove sonic, and something will still be going on. What is this? The cross-repression between these genes and themselves. Namely, the blue gene will inhibit the expression of the red one. So there is a cross-repression at the level of transcriptional factors, not at the level of the morphogen which induced them. And this will enable what? Okay? This will enable the creation of a sharp boundary, and this sharp boundary means that now I am a red cell and now I am a blue cell. Now I have an identity. So the final identity of a cell, okay, will be determined basically by two elements. First, by the formation of a general field of competence created by the morphogen, which stimulates the neighboring cells to transcribe certain transcription factors and inhibits transcription factors that are characteristic of the antagonistic area. And then by a cross-repressive interaction between these transcription factors and themselves, which will yield different combinations of colors of transcription factors, so that this one will mean floor plate, this one will mean V3 interneurons, and this combination will mean motoneurons. So every cell type is not defined by a single transcription factor, but rather by a code, a combinatorial code of different transcription factors. Okay. It's exactly the same. The same mechanism as this one, but, okay?
No, I meant like there are 4 codes here, right?
There are 3 codes in this case for each type of cell. Which is the upper one? Forget about it. Forget about it. This idea is just to define the ventral thing. Okay? So if you imagine that you have BMP up in the upper part, then obviously you will have a very similar game over there, but this is not the point of the slide. The point of the slide is just to work on what is happening in the ventral part. Imagine a similar mechanism upwards. Right? Okay.
So you mean the boundary is very sharp and it won't like different layers in the field.
Imagine a single cell. Imagine you are a single cell in a field, okay, and you have a neighbor, 2 cells, okay. So if you are in this area over here, okay, these 2 cells will fight with each other. So this will be called NKX 6.1, and this will be called the OLIC2. These are the names of the actual transcription factors. So if I express NKX, I will inhibit in myself the expression of OLIC2. And if I am an OLIC2 positive cell, I will inhibit the expression of, okay, and it's a matter of quantity. The more I have of a transcription factor, the best I will inhibit the other transcription factor from being expressed in the same cell. So now go to a population of cells, because this happens in a population of epithelial cells which do not move. Okay, you have a kind of population effect. And everything is cell autonomous here, and also cell-cell interactions. Okay, maybe next week I'll show you some examples of how a certain gene can cross-regulate, cross-inhibit another gene in a certain cell.
There are also some feedback mechanism or some post mechanism that verifies that the cells were dividing right and like fixes mistakes.
We basically don't know about that, because in lower vertebrates or in lower animals, even in invertebrates, basically, for example, a dorsal root ganglion, which transmit the entire sensory information, is composed in the zebrafish of 3 cells. So it's invariant, it's deterministic. There is almost no cell death there, because if you lose 1 out of 3 cells, you are done. Okay? However, in our system, there is cell death, but not at this stage. At the specification stage, we can see extremely low levels of apoptosis. But we don't think that there is a mechanism of correction at this particular time point. Having said that, we know from other systems, not from the nervous system, but from the mesodermal system, that cells which were not properly specified, namely, if you happen to have here a red cell within the blue territory, it will not necessarily die, it will move to the red territory. And why so? Because when you are specified as a certain cell, you are also going, for example, in the nervous system, to express a certain set of adhesion molecules. And adhesion molecules act in what we call the homophilic mechanism. Namely, Encadirin will speak to encadirin. Ecadirin will speak to ecadirin. So if you are an encadirin expressing cell within an ecadirin domain, you will look for your friend. So you will move until you connect to another encadirin expressing cell. So the fact that you express a certain transcription factor is only the beginning of a chain of events that will create your final identity. So corrections are not only made by cell death, they are made also by movement, very small movements. For example, in the chick embryo, this entire system, give me a number. How big can it be in a 2-3 day old embryo? Do you have an idea? Talk to me in microns. This is 100 microns. Okay, 100 to 200 microns of height. Okay, each cell is between 5 and 10 microns. How many cells do you have there? Okay, and obviously it multiplies itself with time. So we are talking about decisions within a territory which doesn't have that many cells, has thousands of cells, right? Because it's a tube. Okay, so this is the idea, of course, and this is how you can see the territories. I brought it just to give you the image. Look at this. You have a pattern that has formed here with the actual true expression of NKX 6.1, OLIC2, PAC6, PAC7, and so on and so forth. And this is just because we don't have enough colors to perform more stainings within the single one. So this is the way in which a very, very clear and homogeneous epithelium creates different domains. Okay. I would like to spend a little bit more of time with this sonic molecule to complicate it a little bit more. And to say that patterning of the nervous system not only depends on sonic, that comes from the notochord and floor plate directly to the tube, it also depends on sonic coming from the adjacent mesoderm. Okay, and to this I need to define what mesoderm is. So first of all, let us look at this picture. This is the neural tube. We have removed the epidermis, okay? We have peeled it off. This is a scanning electron micrograph. So what you see here is the neural tube, and these are crest cells. These are those cells which will generate the peripheral nervous system. We will talk about them maybe next week because I see that I will not have enough time today. And this is the mesoderm. Now the mesoderm is cells which are created as mesenchyme by the process that we call gastrulation. This is the tail of the embryo, and here would be the head. And with time, there is an oscillator, a timer, that creates every couple of hours from this area, creates these little cubes that are called somites. And everything goes like this. This is a mesenchymal to epithelial transition, and then the somites are going to undergo an epithelial to mesenchymal, mesenchymal to epithelial, and by these sequential events, they are going to form muscle, vertebra, dermis, blood vessels, and all that stuff. So this is not necessarily important for us. What is important is that there is an intimate crosstalk between the mesoderm on both sides of the neural tube and the nervous tissue itself. So here, just to have the idea of how it looks like, imagine your spinal cord. Okay, you have the spinal cord inside, and our spinal cord is embedded within the vertebral column, right? The vertebral column comes from the somites, and all the muscles here and all the muscles of your limbs come from the somites. Let me see if this works. Just to show you how the process of somatogenesis takes place. Okay, so there is an elongation of the body, and there is a continuous process of segmentation. This is how the embryo is formed. Then if we look at a single somite, it is an epithelium, but then it loses epitheliality, EMT. This will be the vertebra. And all this is going to be the muscles. So you see an initial muscle, this is called the dermomyotome, and this will contribute further to the formation of your vertebral muscles. Okay, via different lips, etc., etc. This is not important for us anymore. Okay, so many people have contributed over time to the concept that there are intimate interactions between the tube and the mesoderm. So we know that BMP from this area and wind affect the formation of the medial portion of the somites. We know that BMP and noggin, this is our work from many years ago, are the master regulators for the emigration of neural crest cells and the formation of the peripheral nervous system. We know that a molecule like delta notch, a very interesting system in development, is important for the development of muscle, and it comes from the tube and via the crest cells. And we know that there are additional interactions from this dermomyotome back onto the neural tube that determine the timing of the emigration of neural progenitors of the crest. So we know, it's not important that you remember that, but we know that there is a very intimate conversation between the two. And what I would like to argue now is that sonic hedgehog plays a role also in this interaction. And let me show you already an experimental system, a true experiment, okay? In the chick embryo, what you can do is electroporate. You can put the embryo, inject either the somites, like it has been done here, or the neural tube, with DNA that encodes for certain plasmids. And you expose the embryo to an electrical chair. Okay, you take the egg, you put electrodes, DNA is negatively charged, so within an electrical field it will run to the positive pole. And by this it will enter the cells. Electroporation creates pores, little holes in the cells, and DNA will enter. And then DNA will get transcribed, translated into protein. So if here you take a control AFP, this is green fluorescent protein, and you electroporate here half of the embryo, this is the mesoderm, what you see is normal expression of a dorsal gene like PAC7, normal expression of a ventral gene which is called HB9 and is a motoneuron marker. Can we have the lights dimmed for a second? Thank you. This is a motoneuron marker, the red one. And here we can see that, so this is symmetrical. But here we can see that if we electroporate this mesoderm here with one of the enemies of sonic hedgehog, you remember hedgehog interacting protein, it binds to hedgehog like the receptor, but does nothing with it. It acts as a sink. So if you do this, look what happens. There is a dorsalization of the tube because PAC7, the dorsal marker, is now much longer than in the other side. So you have lost sonic, you have lost ventral properties. And you have much reduced motoneuron numbers, okay, than in the control side. So what have we done here? What we have done here is we have basically reduced sonic hedgehog, not within the tube, but within the mesoderm. And this would mean that there is sonic hedgehog in the mesoderm, and that sonic hedgehog from the mesoderm is able to affect neurogenesis, the development of the neural tube. Now, it appears that sonic hedgehog does the following. If I draw the scheme here, it appears that if this is the neural tube, this is the floor plate, and this is the notochord, what sonic hedgehog does is to diffuse to the dorsal neural tube, but it also diffuses in a radial manner, like a sun, okay? Towards the endoderm here, and eventually there it will be involved in endoderm development, pancreas development, for example, but also to the mesoderm. And in the mesoderm, it will be involved in development of the vertebra and of muscle. So, in the absence of sonic, muscle will be damaged. In the presence of sonic, muscle will be well differentiated. Why is this important? Because you have a molecule which is essential for the development of motor neurons, which will then innervate muscle. So it makes sense that a single molecule is able to integrate the development of two interacting systems, like motor neurons on the one hand and muscle on the other, which is the target. So let me show you how basically, if you is it there? How if you inhibit sonic activity in the neural tube, you are going to affect muscle as well. So, for example, look here. This is control, and this is the hemineural tube, which is electroporated. Look, the muscle is blue. You have very little muscle here compared to here. You have the same amount of muscle here and here, but look, this is hip. The muscle and the progenitor of the muscle are very, very affected in this side, and they are normal in the contralateral side. Another example, normal development of muscle and very disturbed development of muscle. So sonic from the tube is able to affect myogenesis, and sonic from the mesoderm is able to affect neurogenesis. So this is a new, basically a new finding, which implicates sonic not only as a local morphogen, but as a molecule which is able to get distributed from one side to the other, from this central tissue to everywhere, and from there basically affect the development of the adjacent structures. Let me skip this and go to a more general part. So let's forget a little bit about sonic and talk during the 15 minutes that are left about different characteristics of the neuroepithelium. So what I told you up to now, and again, you recognize already the system: neural tube, notochord, somite, ectoderm. Okay? Are we okay? Now, what is the neural tube at this stage? I told you in one word earlier that the neural tube is composed of pseudostratified cells. Anybody knows what this is? Okay. So, a pseudostratified epithelium means that you have one single cell spanning the entire extent or the entire thickness of the epithelium. But look at this. This cell has its nucleus here. The nucleus is where the cell is thicker. So this cell has its nucleus here. This one is here. This one is there. This one is there. The same for the somite. What does this mean? Let me show it and explain it to you with this cartoon. A pseudostratified epithelium means that you have one cell thickness, but that the nucleus of the cell is localized at different heights, at different positions. And these different positions of the nucleus determine where they are encountered within the cell cycle of the cell. You know that the cell cycle has 4 phases. Mitosis, M, G1, G2, and S. The phase in which the cell synthesizes DNA. Now it appears that within a pseudostratified epithelium, the position of the nucleus, the cell is always there with its processes, but the nucleus will be modified based on its location within the cell cycle. So, a cell at G1 will have the nucleus in a central position. A cell that approaches the synthetic phase will be located basally in the outer part of the neural tube. And then when it goes through G2, it will approach the apical part, the lumen, the core of the neural tube. Okay, and then it will undergo mitosis in the luminal part. So let me see if I can spot for you some of these things here. No, but mitosis will take place here, and S phases will take place here. These cells are in the S phase, for example, okay? Because they have their nuclei in a position which is basal. So the cell will undergo mitosis in the apical domain. It can undergo mitosis in two ways, and I'm coming to the issue of cell fate decisions versus growth. It can undergo symmetric mitosis and asymmetric mitosis. Now let's look at this. Symmetric mitosis is when the cell that reached the apical domain undergoes a cell division which will leave the two cells in the epithelium as progenitor cells. Namely, what we have done with this type of mitosis is we have multiplied our cells, but we haven't taken any decision about our fate. These two cells will continue undergoing this process, which is called interkinetic nuclear migration. Okay? So, and these two cells will generate, for example, two progenitors. But there is another type of cell division, which is called asymmetric. And look at it. The plane of cell division now will be perpendicular to the previous one. And this perpendicular plane of cell division will create two cells, one which will remain in contact with the apical domain of the epithelium, and one which will be far from it. So it appears that in most cases, the apical cell will regenerate a progenitor that will again go through interkinetic nuclear migration and another round of mitosis, whereas the basal cell will do what? Migrate and differentiate into a neuron, for example. So this is asymmetric cell division. So symmetric cell division will create two identical daughter cells, and asymmetric cell division will create two distinct daughter cells, one which normally continues the round of cell divisions to account for the growth of the system, and another one which will generate already, for example, a neuron. And we know, based on very famous works by Sue McConnell years ago, that at the beginning the neuroepithelium of the central nervous system undergoes symmetric divisions, and this is why. Why would you undergo massively symmetric divisions? Because you need a lot of cells. You have to grow to a certain point until you begin differentiating, right? Because a neuron will withdraw from the cell cycle. A neuron will eventually become postmitotic. So a neuron will not proliferate. So you need many, many progenitors. The proliferation is done at the progenitor level, not at the neuronal level. And then at a certain time in development of the cortex, she used to work on the cortex, the cells will shift to most of them becoming mitotically asymmetric. And this will be the massive time of neurogenesis. Okay.
We know how this asymmetry happens?
Oh yes. Oh yes. You don't want to know now, right? There are a lot of molecules of the LGL and LGL family. These are motor molecules which are located in the cell and which change the plane in which the chromosomes are arranged during mitosis. Okay, because the chromosomes will separate and are attached by microtubules, and these are attached to different molecules, and they are attached to this area of the cell versus this area. It's quite complicated, a field in biology called the cell cycle field, etc. And there is another patent which I'll show you in a minute. Okay? And this patent says that there are, let me just, look at this. And you tell me what happens here. What is this? Anybody help? Obviously, this is a cell undergoing mitosis, right? But which kind of mitosis is this? Symmetric or asymmetric? Asymmetric, correct. Why? Right. So look, this would answer a little bit your question, but the Hatgadia, okay, it's not that simple, because at a certain point in development during the phase in which this cell will undergo migration, there is a marker. We don't know which it is, right? It's not important right now, that will become segregated to one pole of the cell. So the cell must be different in different areas, right? Because it's going to segregate its chromosomes. So once this white marker becomes expressed in one place and not in the other, the end result of this mitotic event will be the formation of two different cells. And this, Rabotai, is creating heterogeneity. This means that you begin with an epithelium that is homogeneous and you end up with two different cells. And this is how the different cells in your body are created. So this is how, okay, in a very, very simplified, oversimplified way. Okay, I'm not entering the details of it, but I assume that each one of the slides I showed to you is basically the basis of a whole field of research. Okay? So this you have to take into account. Okay, so one way by which neurons or neuroblasts will reach their target is by migrating through radial glial cells. These are progenitors, again, that span the entire thickness of the epithelium, but at a certain point will serve as a scaffold for these neurons to migrate on top of them, okay? So the neurons will use the scaffold of radial glial cells in order to migrate, let's say, from basal to apical and form, for example, the different layers of the cortex. So this is one of the main mechanisms for cell migration in the cortex. Now, what happens to the radial glial cells when all this is over? What happens to them? What? They become neurons. Most of them become neurons, right? This is very beautiful data, even in adulthood. Even in adulthood, there is a possibility for these stem cells, which are called radial glial-like cells, to become neurons. Right? Okay?
They have to go back to, I guess, previous state and then become neurons?
No. Radial glial cells are not fully differentiated cells. Okay, so this is one idea. So this is cell migration. So now let me give you, hotspots. If they can proliferate. Good question. I don't know. Good question. I should inquire into that. In the adult they don't. In the embryo, I suppose that they are quite quiescent, but maybe they have the capability. Good question. What is this? What is this mechanism that we see here? Once you have a neuron, the neuron grows neurites. It will grow an axon, it will grow dendrites. Now these cells will have to connect to each other, and they do so by processes called axon guidance. Namely, how do we know that a certain neuron will get to its target? For example, this is an interneuron localized in the dorsal portion of the spinal cord. It sends a neurite. This neurite reaches the floor plate, crosses the floor plate to the contralateral side, and goes to the brain. You see it here. Neurons go to the floor plate, cross, and go to the brain. How does the neuron know what is the pathway to be followed by its neurite? How does it know where to reach the brain? Okay, I told you these are hotspots. I'm not going to give you all the details. Again, this is a field of research. So there are processes called axon guidance. So it appears that the very same gradient of sonic that affects specification, you have gradients of other molecules called netrins. So netrins are expressed here. And netrins create a gradient towards the dorsal portion of the tube. And once interneurons have differentiated, they will go, במעלה הגרדיאנט, they will go towards the highest levels of netrin, arrive into this area, and then they will change the receptors which is very funny. They will not respond to netrin any longer. They will begin responding to another factor that is located here, which is a repellent. And once they find the repellent here, they will leave and go towards the brain. So this is one mechanism for axon guidance. There are gradients of attractants, gradients of chemorepellents, and they are changes in the repertoire of receptors, which are absolutely dynamic. And this is the way a neuron that has specified, differentiated, send an axon will finally find its target. So they are very, very local cues that a certain axon will find throughout its life. And there is a very big difference between the first axon, or the first set of axons, that we call pioneer axons, and the rest in the population. Because what do the rest do? They follow the first. It's like people. Only pioneers do the real job, and the rest are just followers. Okay, this happens a lot in cell migration in development, and it is almost a rule in guidance processes in neurogenesis. So the question is always, what are the guidance cues that are going to be followed by the pioneer fibers emanating from a certain initial subset of neurons? And the rest just follows by cell adhesion. Okay? So this is the other element that I wanted to point out because it is very important. Let's keep all the crest issues today because we are going to devote it to next week. And in the 2 minutes that remain, let me just give you the example of apoptosis that we began discussing in the first talk. I told you that apoptosis, based on the neurotrophic hypothesis, depends on the size of the target organ that you have to innervate. And the beautiful classical examples, okay, are the following. You perform the removal of a limb, or you can transplant in an ectopic place an extra limb, or you can paralyze activity, and this is basically what you get. So if you perform an ablation of a limb, this is the hind limb, you get less motoneurons than on the other side. If you put an extra limb, you get an extra survival of cells, and basically if you paralyze the system, again you get less motoneurons than normal. What does this mean? First, that neuronal activity is needed for controlling cell number, in this case electrical activity. It means that the size of the target will determine the extent of survival or death. And this is the basis for the neurotrophic hypothesis. Now, to this end, we need to understand synaptogenesis. And in fact, these are neurons, all of them coming to the target. The target is a source of neurotrophic factors. And when the supply of neurotrophic factors becomes limited, based on the size of the organ that you innervate, then you get cell death. Only 2 out of the 4 cells that initially innervated the target will eventually survive. And just a picture to show you this. You see here a neuromuscular junction. These are fibers. These are acetylcholine receptors in aggregates on the muscle. These are muscle fibers, beautiful muscle fibers. Okay, and you can see the process of synapse elimination. You see the initial innervation of the system, okay, and you see how most of the synapses have been eliminated during a normal process of elimination. This is not disease. This is normal life. Okay? Any questions? How many? I don't know, but I can guess. Okay, if this would be a chick embryo, which I don't know, this would be around day 10 or 11 out of 20 days of incubation in the egg. If this would be a mouse embryo, this would be around day 15 of development. Okay? mid-gestation, more or less. In the mouse embryo, it's a little bit later than mid-gestation. The mouse embryo is born by 21 days, right? Something like that? Okay. So we have spoken here about several processes, and I'm going to wrap up everything I told you very briefly, just a taste. We talked about the nervous system being an essential part of the context of the embryo. We talked about axial level identities generated by Hox genes. We talked about evolutionary conservation at the early stages. We talked about processes like cell specification and the generation of an identity that are created by gradients of different morphogens, by concentration, time of exposure, which bring about the formation or the expression of different transcription factors that on their own have cross-inhibitory interactions which enable to sharpen a boundary and create a specific fate. We talked about adhesion molecules that are also downstream this process, about the neural tube undergoing symmetric cell divisions to account for growth and asymmetric cell divisions to account for the formation of heterogeneity. Once the cell is differentiated, it will send the neurite. The neurite will grow and has to be guided by specific cues of the environment to its correct targets. And when it gets to the target, it will measure the amount of factors for survival, and based on that, it will survive or die. So we are a redundant system. We create many more things than we actually need, and we adapt to the environment for final shaping. Questions? Okay, if not, next week I hope we will talk a little bit about the peripheral nerves. Okay? תודה רבה. העסק לא נוצר שם, העסק נוצר הרבה קודם.